Well, I want to thank the organizers, particularly James Taylor, for giving me this opportunity to speak to you, and also, more importantly for me, to get the opportunity to meet all of you and hear the wonderful things that you're doing. Actually, James, you don't know this, but if you hadn't asked me to speak, I would have come anyway, because this is an important meeting for me to learn, because I'm not a nanoparticle person. I'm a biologist and a physician. This is where I work in the Center for the Houston Methodist Research Institute. I direct the Center for Cardiovascular Regeneration. I'm the chair of the Department of Cardiovascular Sciences there. I came to the Texas Medical Center from Stanford in 2013 because it's a very exciting place. We've got a CEO who is a nanomedicine doc. And actually, Mauro Ferrari is arguably the father of nanomedicine, and we've got a small army of nanoscientists there to work with. James asked me to talk about the application of nanotherapeutics to cardiovascular disease, and I'm going to focus on a specific cardiovascular disease. Hutchinson-Gilford progeria syndrome. This is a rare disease of accelerated vascular aging, but it has broad relevance to all of us because it mimics the major cause of morbidity and mortality in the world, and that is atherosclerosis. We've developed a new approach to HGPS that I think looks very promising, but we need a way to deliver it, and that's why I became interested in this field and what you all are doing. And I'll talk about our preliminary work in developing nanovectors for our nanotherapeutic. Let me introduce you to Dr. Leslie Gordon. About 20 years ago, her son Sam was born with progeria. Dr. Gordon is a professor at Brown University, and she was stunned to find out that no one knew what was the mechanism of progeria. She convinced Francis Collins of the NIH to the director of the National Institutes of Health to pursue the genetic underpinnings of progeria, Hutchinson-Gilford progeria syndrome. And he actually discovered that it was a problem with lamin A, a mutation in lamin A that creates this abnormal protein progerin that I'll talk about in just a moment. I show this picture. These are children from the first clinical trial for a new therapy for progeria. And I show it because it documents the classic phenotype of these kids. The diagnosis is usually not made at birth. It's usually made at the age of 2 to 3 when the kids are dropping off the growth curve. They have failure to thrive. Their weight is very low. They have reduced subcutaneous tissue. They have alopecia. They begin to get osteoporosis, all the manifestations of accelerated aging. What kills these children at the age of 13, 14, 15 years is when they die, is this disease. This is a vascular disease. It's very similar to atherosclerosis in adults. These are coronary arteries from children that had progeria and succumbed to myocardial infarction. These coronaries, to me, as a cardiologist, look like those of a sick 80-year-old person, but they're from these children who have accelerated aging of the mesodermal tissues. Ironically, or tragically, the brain is unaffected, so the children are bright, alert, they want to be kids, they have all the joy and joie de vivre of children, all the playfulness of children, and it's really tragic that they succumb to this disease at the age of 13, 14, or 15. And it's really a model of accelerated atherosclerosis. This is a disease, the major cause worldwide now of morbidity and mortality, causing stroke, heart attack, renal failure, aneurysm, mesenteric ischemia, peripheral arterial disease, and for us, this is something we deal with every day as cardiologists, and we're always looking for new approaches. And something that's interested me as a potential approach for atherosclerosis for 30 years has been the possibility that we could restore the health of the endothelium. You are only as old as your endothelium. The endothelium is the lining of all our blood vessels. It's a diaphanous film of tissue, a very thin, fragile film, one layer thick, one cell layer thick, but it exerts tremendous control over vascular structure, vascular tone, and interaction of the blood vessel with circulating blood elements. Normally, our endothelium is like Teflon. It's generating products that cause vasodilation, that cause the blood to flow smoothly, that reduces vascular thickening. One of those that I've studied for quite a few years now is nitric oxide. Endothelium-derived nitric oxide has all of those effects. In 1998, 3 American scientists won the Nobel Prize for the discovery of nitric oxide as a vasodilator. And since then, it's been also shown to inhibit platelet aggregation, inflammation, vascular inflammation, smooth muscle hypertrophy. A dysfunction of the endothelium, and characterized by loss of that ability to generate nitric oxide, occurs with aging. We see in front of you there a picture of normal endothelium. It's responsive to shear stress in the blood vessel. The endothelial cells line up with shear stress, with blood flow. Aged endothelium looks more like cobblestone paving. This is actually endothelium that's in this flow of bifurcation. So at bifurcations, the endothelium looks a little bit more like this. It's aging faster, it's not aligned with flow, and it's producing adhesion molecules and chemokines that accelerate the atherosclerotic process. Back to progeria for a moment. As I mentioned, there's a mutation in lamin A that causes aberrant splicing of the protein. Normally, lamin A is farnesylated, and by virtue of that farnesylation, it associates with the nuclear envelope. But then the farnesyl group is cleaved, and the lamin A is left in position near the nuclear envelope. But the abnormal progerin, the cleavage site has been deleted. So the farnesylation cannot be removed, and the progerin sticks to the nuclear envelope. As a result of this progerin accumulation, or I should say it's believed as a result of this progerin accumulation, you get distortion of the nuclear envelope. As pictured, the nuclear envelope becomes lobulated. The whole genomic architecture of the nucleus is distorted, and gene expression changes, and you get a SASP, senescence-associated secretory phenotype, in cells from these children. And that's what is believed to cause the accelerated aging. The only drug that's been shown to be useful for these children is an agent that inhibits farnesyl production, and that is lanafarnib. The farnesyl transferase inhibitor prevents the farnesyl group from being added to the protein, and that is somewhat effective. It improves, shown here, the pulse wave velocity. Pulse wave velocity is a measure of vascular stiffness. The higher the pulse wave velocity, the stiffer the vessels. And at the end of therapy, these children that were treated with the agent had somewhat more compliant vessels. Furthermore, although it was a small study, the children seemed to live somewhat longer than expected. As I said, they normally die at the age of 13 from a heart attack or stroke. These children lived to about 15 years of age. Clearly, though, it's an unmet need. We need something better. And we're studying this because the kids really need something better, and also it's broadly relevant to all of us because this is a disease that does affect us all as we get older. By the way, even though a normal individual doesn't have this mutation, abnormal splicing of lamin A does occur in all of us to a certain extent. As we age, progerin actually does accumulate in our nucleus as well. So I'm going to tell you about what we've done to develop a new therapy for HGPS. As I mentioned, I'm directing a Center for Vascular Regeneration. I've had a long-standing interest in telomerase. I don't have much chance to review, I don't have the time to review all of that past work. But we noted that, and it's been noted by others, that the children with this condition have rapid telomere erosion. That's part of the senescent phenotype. Telomere erosion occurs as we get older, and it's due to the end replication problem. And also, in addition to that, stress, intracellular reactive oxygen species also cause telomere erosion. So with the telomere erosion that is accelerated in these children, we asked the question, could we rejuvenate the cells? Could we treat these children's cells with telomerase and improve, extend the telomere? And if, in doing so, could that have a beneficial effect? The idea being that by the time these kids are recognized as having the disease, they've already had significant telomere erosion. To the extent that the telomere length generates a limit on cell replication, to the extent that telomere length predicts senescent phenotype, we thought we could extend the telomere and improve these children potentially. I think you're all familiar with this idea that telomeres are basically the tip of the chromosome, the cap on the chromosome, prevents a DNA injury response. It's basically multiple repeats in humans, on average of 8 to 10 kilobase of these hexanucleotide repeats. And every time the cell divides, there's this end replication problem where you lose a bit of the lagging strand, and the telomerase replaces that loss. Telomerase is inactivated in somatic cells. Telomerase, the enzyme, is only present at the embryonic stem cell stage or in induced pluripotent stem cells, and that's why these cells have the ability to replicate indefinitely. Embryonic stem cells, an embryonic stem cell, the zygote, the human zygote, that has to become a trillion cells to become a human individual. So a lot of divisions in an embryonic stem cell, there's a reason for it to express telomerase. Telomerase is reactivated in iPSCs. It's also activated in what other cell type? Cancers, right? Cancer is another. 85% of cancers have reactivation of telomerase. So that gives them their ability to replicate indefinitely. Our thought was, we initially had, working with Giron many years ago, we had shown that we could rejuvenate senescent endothelium with retroviral vector that integrated telomerase into the endothelial cell. But that's not a really good solution because you have integration of telomerase, and essentially you've telomerized, you've immortalized the cell. So we were interested to do that because we and others had shown that aged endothelial cells have short telomeres, so these are normal human endothelial cells. Chang and Hurley showed quite a few years ago, about 15 years ago, that endothelial cells at the bifurcation of a blood vessel also have short telomeres. Endothelial cells are turning over faster at bifurcations, branches, and bends in the blood vessel. And that's where the lesions occur in atherosclerosis, at those sites of focal senescence. So here's the study I mentioned that we did with Giron quite a few years ago. And I won't go into great detail about the study, but essentially what it shows you is that the parental cells, these are the earlier passage cells, can produce nitric oxide. The senescent cells are not able to do that, even with stimulation. But when those senescent cells are treated with H-TERT, they can now generate nitric oxide. As I mentioned, that's a key homeostatic factor for the endothelium. But it's not a great solution. So many years later, we revisited this and said, well, maybe we can just use RNA encoding telomerase. And obviously, it won't immortalize the cells. It doesn't alter the cell genome. And we decided to revisit this because we had already developed, and I'll talk about it in a moment, GMP processes and procedures for synthesizing, purifying, qualifying RNA as a result of the funding that we've gotten from the state government to do this for the cancer community in Texas. So we were able to generate the H-TERT telomerase and show, in fact, that we could extend telomeres. This is work from the progeria studies that we've done. Whoops, sorry about that. Let me go back. Up in the right-hand corner, you can see the increase in telomere length by mmQPCR that we were able to gain with 3 transfections of the fibroblasts, progeria, that derive from these children. And let me show you the effect that the therapy had on proliferation. In the blue and dark line at the bottom of the, these progeria cells proliferate very poorly. Over time, you can see that there's maybe a doubling of the cells, and then they start to fade away. Those were the control cells. We also did in blue, we transfected the cells with catalytically inactive telomerase. In the black solid line are those the response of the progeria cells treated with RNA telomerase. And you can see there's a dramatic improvement in the replicative capacity of these cells. And we've carried this out further, and they do plateau. They will age. So these cells are not immortalized. Furthermore, we've actually measured telomerase activity and the persistence of the RNA. And as you can imagine, the RNA after the transfection only persists for a few hours. The telomerase protein that's generated only persists for about 48 to 72 hours. And the surprising aspect of this was that we got clinically significant telomere extension just with a transient transfection. And that is what allowed us to get the intellectual property when I was at Stanford on this approach. We've compared the beta-gal expression that we get in control cells and those that are treated with lonafarnib, H-TERT, H-TERT plus lonafarnib. Lonafarnib is one drug that's being approved for use in these patients, and it does maybe a little bit to reduce beta-gal expression. It's a senescence marker, but the transfection with H-TERT is much better. Because of time, I'm not going to show all of the other data. It's going to be published soon. But we were able to show that in addition to improvements in replicative capacity, the function of the fibroblast was improved. There was a reduction in the generation of reactive oxygen species. There was a reduction in the production of senescent chemokines, inflammatory cytokines. And what was surprising was that there was an improvement in the nuclear envelope. This is improvement that we see is not limited to progeria cells. These are normal cells, fibroblasts from that we've made senescent and with multiple passages. And you can see the benefit that we get with 1, 2, or 3 transfections of the RNA telomerase. These are normal human fibroblasts that have been aged. I'm going to skip over this because I've summarized what we've done with the RNA telomerase, and what we're now have to do is to generate GMP RNA if we want to really bring this to the clinic. I'm a physician. I want to see this scientific development get translated into something useful. So we were fortunate to get funding from the state government to develop a clinical grade RNA for the cancer community. So we're up here in the Research Institute. We have on the 12th floor a GMP facility. It's small, but it's good for GMP manufacture for phase 1 and 2A studies. And we're generating the RNA with proprietary approaches. You can't use kits if you want to put the RNA into the clinic. You have to develop your own synthetic procedures and methods to purify the RNA and to qualify it. But now what we need to do is to develop a way to target the vasculature. And that's why I'm here at this meeting, and that's why I've contacted James Taylor previously to develop methods, to work with you to find methods to get this therapy to children. I'm fortunate to be in a research institute that's led by the CEO, Mauro Ferrari, who's a nanoscientist, and we're working with several of his colleagues to develop the different nanoparticle approaches. I'm going to introduce Roberto Molinari to come up and talk a little bit about the approaches that we're doing, because Roberto is actually a nanoscientist and I'm not. So this is Dr. Roberto Molinari, and he's going to talk about his work with us on the cytopods.
Thank you. I can. Hi, everybody. This was very unexpected, so it's good to meet friends at conferences. So as John was said, we started this collaboration in Houston because his lab was looking for delivery strategies for Thanks. So delivery strategies for messenger RNAs. You have already heard of the limitations and what are the advantages of using nanoparticles for delivery. And actually what we developed in Houston and was published last year in Nature Materials is a biomimetic approach. So we developed liposome-like biomimeric nanoparticles having the membrane proteins of leukocytes incorporated into the lipid bilayer of a liposome. Why we thought about these strategies? As you know, leukocytes are the main effector of our immune system, and they have natural capacities of escaping the reticulum endothelial system, so the clearance by the res organs, and at the same time they have natural targeting towards the inflamed endothelium. And as John was mentioning before, the endothelium of this patients is activated, that is overexpresses cell adhesion molecules like ICAM1, VCAM1. So we developed these particles, we characterized them, and we showed that their surface expresses the integrins typical of the leukocytes like LFA1, like VLA4, like PSJ1, and we have demonstrated in vivo, in vitro, and in vivo how these particles can escape the clearance by the immune system and at the same time can accumulate at the site of the inflammation. In particular, the reason why we are here is that we have just developed a protocol to develop these biomimeric nanoparticles using the nanoassembler system. I cannot disclose much. There is a paper under revision, hopefully will go well, and hopefully you will hear more. But the take-home message of this approach is that we can use biomimeric nanoparticles formulated using the nanoassembler and encapsulating modified messenger RNA in order to target specifically the inflamed endothelium. And I think, okay, here's some data of characterization I was mentioning before. So these nanoparticles are 120 nanometer in size, and as you can see from the cryo-EM images, they have the typical liposomal shape, but you will find more on our publication. Thank you very much.
Much unplanned, but I saw Roberto in the audience, so I asked him to come up and talk about his work. So thank you, Roberto, for that. I want to conclude just by saying that there's a lot of cardiopulmonary applications. Obviously, if we can develop vascular targeting for RNA telomerase. One of the things the low-hanging fruit for us is enhancement of other cell therapies, and there we actually don't need a nanoparticle necessarily. We were actually successful using nucleofection for cell therapies, supercharging other people's cell therapies, like CAR T cell therapies or regulatory T lymphocyte therapies. And there's a lot of potential other applications for an RNA telomerase. I want to end by thanking the Costas family for the support they've given us for nanocardiology and also the team that does the work. Thank you very much for your attention.
Questions?
Go ahead, James. I'll repeat it so everyone can hear.
So awesome talk. Thank you. That was fantastic. Question is, do you think that you'll need to target the entire vasculature, or are there strategies to target the stem cells or something like that?
Well, so that's a great question, James. For this application, these children have diffuse abnormality of their vasculature with accelerated vascular aging. So I'm not certain that we need to specifically target any particular vasculature because I anticipate that it's diffusely diseased. That being said, the beauty of these cytopods that are basically nanoparticles that are cloaked in the membrane proteins of leukocytes is that they go to sites of inflammation. That's in Molinaro's Nature Materials paper. Well, the senescent endothelium is like an inflamed endothelium. It expresses adhesion molecules, VCAM, et cetera. And so the leukosomes we think are probably going to end up there, the cytopods. Yeah. Yes?
So another question. You had mentioned there on the slide that when you add the telomerase mRNA, there's no evidence of any sort of oncogenic influence. But how confident are you that that would be the case, especially if this was going to be used more broadly as well?
This is a question that gets regularly raised. It's an important question because we are giving an exogenous telomerase, and anyone that understands oncology recognizes that that's the problem with cancer stem cells. They've reactivated telomerase, 85% of cancers. So my response to that is, we obviously have to be alert. We have to have a high index of suspicion. I think it's unlikely because the RNA, of course, is only lasting for a few hours. The telomerase protein itself is only around for 48 to 72 hours. So we are not immortalizing cells. They are transiently extending their telomere, and that's enough to have a clinical effect. So I think it's unlikely that we're going to have an oncogenic effect.
So that gets to my second question, because you said there is evidence of a prolonged effect, but how long have you measured that effect, and how often do you think you'd need to re-administer in order to have a sustained benefit?
Great question. We actually have looked at this, and the benefit goes out for about 3 months, the benefit that we're seeing. That's as far as we followed it out. And we continue to see an increased growth curve at 3 months. I mean, it's starting to plateau, but there's some benefit. And then if we're doing a cell therapy, we would probably transfect 1 to 3 times to extend the telomeres enough to get an improvement in replicative capacity, to get more of the clinical product, and to improve the persistence in vivo. We actually had a paper in Cell Discovery last year showing that we can do that with CAR T cell therapy. We can improve the persistence of the cells in vivo and get a greater anti-tumor effect by telomerizing the CAR T cells before we put them into the animal. This is very highly dependent upon the cell type also, I should say. Some cells are very responsive to this approach and some not so much.
Thank you. Thank you for a great talk.
I don't think this is working. Okay, it is. Hi. Thank you for a great talk. You spoke about CAR T therapies, and recently Novartis is, they're sort of leading the pack. They're making a lot of progress. What do you think are the big challenges in developing CAR T cell therapies for a wide range of diseases?
Well, I'm not an oncologist. I am trying to help them improve the CAR T cell therapy. But I think it's widely recognized that the major problem for CAR T cell therapy is with solid tumors. Great success with B-cell leukemia, not so much with solid tumors. I'm working with some oncologist, Malcolm Brenner, for example, at Baylor, and we're excited about the hypothesis, this has to be tested, that solid tumors may become more amenable to CAR T cell therapy if we can allow those cells to persist for longer. So maybe our application will be useful there. Thank you.